Well, welcome. Thank you, and for joining us for our Skype. I love the Underwood typewriter in the background.
Oh, I see that.
If that thing could talk, right? Well, this is our small meeting of the club, and we look forward to talking to you about Crest today. We have 2 biopsych majors here, master's students, and 3 neuroscience masters, and 2 pre-med students as well. So when we were chatting beforehand, we've been trying to set up for this this meeting, and we're fairly unfamiliar with Crest. It's a keen interest of mine, but it's a bit of a mystery. Can you explain a little bit about your perspective on Crest?
Sure. Well, first of all, maybe I should tell you how I got started in it. I started as a graduate student, probably just a little bit older than you guys are now, and I actually majored in biophysics. I thought I wanted to be a physicist, but I couldn't quite decide, and so I went to grad school in that area. And because I had most of my background was in physics and chemistry, I had very little biology. So as a graduate student, I had to catch up. And I took a developmental biology course, and I had a very good teacher, and he went through the neural crest. And you know how you just have this epiphany, and you say, "Oh, that's so cool. That's what I want to work on." That's sort of what hit me. And shortly after that, there's this woman who's now a very famous, older scientist. You know, she's sort of the grande dame of the field. Her name is Marianne. She came and gave a seminar where I was a graduate student, and I met her. And I was just absolutely blown away. I thought I wanted to be her. And I actually presented we had to present sort of do a thesis project on somebody else's work as first-year students, and I did it on her papers. And so I really read her papers like a Bible almost. So I'm not religious, but I still and I just really got to know her papers, and she became somebody who I wanted to emulate. So that had a lot to do with it.
So she was a mentor to you?
No, not at all. Actually, I only met her one
vicariously.
time. And the funny thing is, after that, when I got into the field, I was a bit of a competitor, but now she's, I just spent last May, she invited me to become I was a professor at the College de France for a month. So they gave me an apartment, and I gave a bunch of lectures, and it was at her invitation, because now I've become sort of her protege in a way. So it's been really gratifying.
With invitations to France, how nice is that?
So anyway, that's how I got involved in the whole thing. And it just became clear to me that I was fascinated by this particular topic, and I've been working on it really ever since.
Well, you're a pioneer in the field, no doubt. So can you explain it? So they call the neural crest the 4th germ layer. Can you explain what that?
Yeah, so the reason it's called that is, the neural crest is a vertebrate-specific structure, and we tend to be very focused on vertebrates because that's what we are. But in fact, in terms of the tree of life, we're a very tiny little branch. And so somewhere in the evolution of animals like us, which are called chordates, because we have a spinal cord-like structure, this cell type was invented. And in fact, I've become very interested in this. We've been studying very primitive vertebrates to try to understand that. In this transition between invertebrate chordates and vertebrates, this new cell type came about. And the interesting thing is that neural crest cells actually took over some of the cell types that had previously been made by other things. So, for example, the neural crest cells make the jaws of the face, they make most of the peripheral nervous system, and they make all of the pigment cells of our body. Sea urchins have perfectly good pigment cells, and yet they are not vertebrates. And other invertebrates have all these other cell types. That they make cartilage, and they make sensory neurons and stuff like that. But all of a sudden, in vertebrates, the neural crest sort of took over all of these things. And what happened in terms of the formation of the neural crest, it started out developing with the rest of the central nervous system. But then, so the way the neural crest develops, in development, have you guys taken developmental biology?
I'm in a comparative neuroanatomy, mammalian neuroanatomy class, but.
Okay, so the way, so I just actually taught this. So the way embryos start out is that they start out as one cell, the fertilized egg. So imagine, you have a fertilized egg, and the information in that egg has to be divided up. So the first thing that happens is that cell divides into lots of little cells. And as a broad generalization, and the embryo then forms like almost like a hollow sphere. Okay, imagine that. And you can think of all those cells as being equivalent to one another. But then what happens is they decide to rearrange. They rearrange into 3 layers, almost like you can think of it as a peanut butter and jelly sandwich. You have bread, you have peanut butter and jelly, and forget the other bread. So the bread is what we're interested in, which is what forms the nervous system. Underneath that is the peanut butter and then the jelly, and those form other things like muscle and gut tissue. But in terms of nervous system development, the action is in the bread. And if you think of the bread as being a plane, in the midline of the bread, see this? What happens is the nervous system is going to form, and those cells become different from the adjacent cells, which will form the skin. Okay? And so what happens is these cells, imagine them being connected here. They begin to roll up and transform from a flat sheet, which is called the neural plate, into a tube that's called the neural tube. And this neural tube, it closes, and it really literally forms a tube like this. But then in the head, this hollow tube begins to expand and forms the brain vesicles. So it forms the forebrain, the midbrain, and the hindbrain. And then further down in the trunk, it forms the spinal cord. So the whole central nervous system comes from this tube. So the neural crest cells in vertebrates start out in this flat neural plate, but they're at the edges between the neural and the non-neural ectoderm on both sides. And as the tube closes, they're at the leading edges of this closing tube. Then the tube actually physically closes, and they start life contained within the central nervous system. Okay, so they look just like any other neuroepithelial cell. But then they do something amazing, and that is they undergo a transition. They lose their connections, and they transform from an epithelium into what are called migratory mesenchymal cells. And then they migrate all over the place. They undergo the most extensive migrations of any embryonic cell type. And depending upon where they end up, they form ganglia of the peripheral nervous system. They form both the sensory and sympathetic and parasympathetic and enteric ganglia. So almost all the peripheral nervous system. They form, if they migrate into the skin, they form the pigment cells. If they migrate into the facial skeleton, they form cartilage and bone. And so they form all these different derivatives. And in fact, the neural crest is what influenced Darwin to form his hypotheses about natural selection, because the beak of the finch, the famous beak of the finch, the reason they were modified so quickly is because these neural crest cells are this new innovation, and they turn out to be very easy to change. You just, if the cells divide a few more times in the beak, you'll get a longer and thinner beak as opposed to a shorter and fatter beak. So over a relatively short evolutionary time, the facial features got modified, and that's what led to this very rapid adaptation.
Fascinating. So they respond to the environment. They respond to the environment in a way.
Yeah, because they're this new cell type, they seem to just respond more rapidly to change.
And there are many ways they differentiate, and they're. Well, I have these questions that we prepared for you. It's a fascinating area of.
It's not so much. Not much of the nervous system, though, unfortunately.
I'm sorry.
They don't really do much in the nervous system. I mean, they contribute to parts of the nervous system, but they do lots of other things.
They're outside. They're the peripheral nervous system. That's where I don't see the relationship between the melanocytes and the rest of the function of neural crest derivatives. I mean, the pigment cells seem to be out in left field as far as.
But there's, just as much as cartilage, and these cells also form, they can form smooth muscle. They can do lots of different things. There's some literature suggesting they can form fat cells. So they're sort of stem cells, and that's probably an easier way to think about it.
Well, that's really interesting, the Darwinian connection, and that it seems to be a response to environmental cues that neural crest derivatives will modify as quickly as they do. especially the beaks, the bird beaks are a good example. So can you talk about placodes and neural crests and the relationship of those two?
So the placodes only form in the head, and they're very similar to neural crest cells, except that unlike neural crest, they don't contribute to things that are non-nervous system. So they don't form pigment cells, they don't form cartilage, but they do. They're thickenings in the ectoderm, so they're never inside the central nervous system. They're just out in that that outside layer, and they also lose their connections with the rest of the ectodermal cells, and they ingress into the embryo, migrate, and then condense to form ganglia. Or they invaginate and form things like the ears or the nose.
Right.
So they contribute to sensory structures and to cranial ganglia.
Fascinating, but they're in the same area as the neural crest cells?
Well,
they
they're only in the head.
Mm-hmm.
Yeah,
so
so
they derive from the same area of the...
No, they're actually lateral to it. So if you go back to my neural plate,
your high-tech...
yeah, my high-tech thing. So the neural plate closes like this, and then the rest of the ectoderm closes over it.
Mm-hmm.
The placodes form really in the ectoderm itself, and they're within that ectodermal layer. What's really interesting about the neurons is it's a case where the neurons actually differentiate and turn on neuronal markers in the ectoderm, and then they begin to migrate. So they differentiate first.
Fascinating. Maggie had a question. What's the cutoff line between neural crest and when they become other tissues?
Oh, that's a really good question. It's pretty semantic. Yeah. So I usually think about it, basically they're constantly changing. So as they migrate, probably their choices in terms of what they can become gets limited. In terms of the peripheral nervous system, what I usually think happens is they sort of stop migrating, they begin to aggregate, and then they differentiate. And really it's that transition between migration and differentiation that makes them lose being a neural crest cell to becoming a neural crest derivative. But there probably are still neural crest stem cells contained within many of the derivatives, because you can isolate, they sort of remain in the, you can take like a ganglion and transplant some of the cells back into a young embryo, and they'll still make some of the normal derivatives. So there are probably some stem cells that remain.
Do you do that in your lab?
Oh yeah, we used to do that a lot.
Sounds like fun. Sounds like a rap sign. Migrate, aggregate, differentiate. We'll see what we can put up to get going there. So can you talk about your single cell injection work? We talked about the lineage and the fate of these cells, but what are the latest findings?
Oh, well, those are actually pretty old experiments. They were done like 20, in fact, my daughter, I remember, was born too early, and so I didn't quite finish the experiment when she was born, so she's acknowledged in the paper as, being quiet because she was in a crib in the lab while we were finishing the experiments. So she's 23,
okay.
So she's older than you guys. So we don't do that very much anymore, mostly because it's really a hard experiment and I can't get any of my students to do it. But basically what we used to do is inject dye into single cells in the embryo and ask what those cells could become. And that's how we showed that cells were the neural crest precursors were multipotent. In principle, you can now do that same experiment in a much simpler and more elegant way, because you can tag the whole embryo with fluorescent dyes, and then you can photoconvert, say you have cells that are expressing a red dye, you can convert it into a green dye in one particular cell by shining light on it, and then you can follow the lineage of those cells. There hasn't been very much done in terms of that kind of lineage analysis recently. Most of my lab right now is working on hardcore molecular stuff. We're interested in what transcription factors are being turned on at particular sites in the embryo, and how different processes in neural crest development are regulated. So some of these lineage type of experiments, which I think are very cool and can give you some really interesting answers, are not being done anymore because they're quite difficult.
Beautiful. Well, we look forward to following